Hello, my name is Sylvain Faugeron, and I will make an overview of the cultivation of Gracilaria in Chile and how the cultivation practice affect the domestication of the seaweed and how it relates to the problem of epiphytism in the farms. The cultivation of Gracilaria in Chile is dominated by small-scale artisanal farms. It's an extensive productive system with farms usually occurring in intertidal environments, like here in the picture. It's at low tide, the cultivators operate their farms by burying some fragments in the sand or in the mud and let it grow. And then after harvesting the biomass, part of the harvest is replanted in the same place to restock and regenerate the farm. It's an artisanal system because it's dominated by a very small scale operation controlled by families or small communities of local fishermen as a complementary activity to fisheries, by the way. So most of the farming operation are done through clonal propagation, which is one of the important aspects I will develop in the presentation. There are some farms that are using ropes also to cultivate, to produce the biomass. Usually it's also through clonal propagation, so they use small fragments that they entangle in the ropes, and then you have ropes with clonal fragments for the production. And more recently, they are using spores that they settle on ropes, on clean ropes, and using therefore a non-clonal method, which is through the settling of spores. This is more recent, but here in this picture, it's a clonal propagation on ropes. As you can see, always also in the intertidal, but there are some farming operations that are operating in the deeper waters through suspended ropes. It's not really common, but it is occurring also in Chile. So this production, extensive production, started very early in the 80s after the collapse of natural population because of over-harvesting. And it promoted a rapid increase in the total biomass produced by the country. But since the year 2000, we could see a relatively regular trend of decaying of the production of these farms. And this decay was attributed to a degradation of the quality of the biomass, which was growing much less, and eventually also because of epiphytic load, which degrade the quality because all the biomass was including also a lot of non-Gracilaria species. And so the price of this product was not really good. And it was contributing to the decay in the total production in the country. So the presentation is really, I will try to explain you some of the consequences of this cultivation practice and try to relate this to the problems of decaying of the production, both in terms of the genetic resources, the impacts of the domestication of the species, and on the epiphyte problem. So to understand the genetic resource in Chile, we have to consider that there is a very low genetic diversity within farms. It is even lower than, it is among the lowest genetic diversity of all seaweed farming that we know so far. And this is due to three main factors. One factor is the fact that it is a Chilean species, growing naturally in Chile, but introduced from New Zealand. It is a natural introduction that, according to our studies on the genetic diversity, we could infer that it came from New Zealand probably at the end of the last glacial period. So we are talking about 15 to 20,000 years ago. It's not very recent. And so it was probably an introduction from the eastern coast of New Zealand, where one genetic variant, which is represented here by this blue circle, is also the same genetic variant that is present and largely dominating in Chile. So it is this variant that arrived to Chile, and then new local variants that are represented by these small light blue dots started to appear through mutations accumulated in Chile. These mutations are different from the ones that are present in New Zealand. So in New Zealand, there is much more diversity, as you can see, compared to Chile, and a strong effect, which we call the founder effect related to the introduction of the species in our country. So this is one important aspect, that the history of the species is relatively recent in Chile and contributes to a low genetic diversity. The second aspect is the fact that there was an over-harvesting of the natural population during the 60s and the 70s that led to nearly a collapse of the natural populations. And this has effects on the distribution of the genetic diversity, sorry. If we compare New Zealand to Chile, as you can see, there are a certain number of alleles that are missing in Chile. And this type of pattern in population genetics is a clear signature of the effect of population collapse. This population collapse remove a large part of the genetic diversity that eventually was present before that. So in New Zealand, there is no harvesting at all. So we compare the two situations, and we can infer that in Chile, there was a strong effect of harvesting. And the third point is, of course, the clonal propagation in farms. If we quantify the number of different genotypes in farms, we see that usually there is a large dominance of a single clone within farms. So this is, here I show you two examples where, for example, in Caldera, 92% of the total biomass of the farm is actually one clone. La Herradura, another farm, is 84% of the biomass that is on one single clone. Of course, this contrasts with the natural population where we see much more genotypic diversity. So there is a low genetic diversity in Chile, and specifically in farms, because of the clonal propagation, but also because of the over-harvesting, and in a more global context, because of the relatively recent history of the species in Chile. So this is an important aspect, and I will develop this idea later on about the impact of the low genetic diversity. When we analyze the distribution of this genetic diversity in Chile, we see that different farms tend to be genetically similar in terms of composition when they belong to the same region, and the regions are different among them. So this is related to the tradition of local cultivators to exchange the material for settling new farms or restocking the farms that were harvested. They usually use biomass from the neighbors to restock the farm once they have completely harvested, for example. So these exchanges are occurring essentially among neighboring farms within regions, and lead to the creation of genetic characteristics of each region within Chile. Another aspect that is also relevant from the population genetics is that we see very little evidence of exchanges between farms and natural populations. This means that farms and natural population are also independent genetic entities, and we interpret this as the fact that the farmers reuse the same biomass that is present within the farms of the region, and never go to the natural population to extract new variants, new genetic variants to reseed or restock their farms. they are operating relatively independent. And this is also contributing to the genetic erosion of the resource through this practice. So this isolation of the farms from the natural diversity, plus the cultivation practice in terms of vegetatively propagating the same biomass constantly, leads to the selection of some traits that are relevant for the production. One important trait is the one that we see here, where in farms we see mostly diploids. Usually farms have between 95 or to 100% of the biomass that is actually diploids, the tetrasporophytes, except one case where it is dominated by female gametophytes. And this contrasts with the natural populations where we find both diploids and haploids, and which is not normal because in natural populations, usually on rocky shores, the individuals are present through the settlement or recruitment of spores, either carpospores or tetraspores. So to have a stable population on rocky shore, you need to complete the sexual life cycle. And this is why we see the two types of organisms in these populations, as opposed to farms where we see only the diploid stage of the life cycle. And also important aspect is that these diploids actually have lost the capacity to produce spores. When we compare to natural population, most of the individuals are sexually mature and are able to liberate spores, and actually they do this almost year round. When we go to farms, there are very few individuals that are producing spores. And even if we bring these individuals to the laboratory and try to induce the release of spores, actually they do not produce even reproductive structures. So they really lost the capacity to invest in spore production. And this is probably the consequence of the selection for the highest growth rates within the farms. The diploids are the ones that have a higher growth rate. They grow faster than the haploids. This has been confirmed also in the laboratory. And the ones that do not invest energy or resources to the production of spores, they are the ones that also have the highest growth rate. So the farmers are selecting for diploids, and they are selecting for vegetative diploids that are no longer capable of producing spores. So these traits, we interpret this as a signature of domestication, that is the consequence of cultivation practice on the evolution of traits that are also relevant for the cultivation itself. So this is the context. The cultivation practice been modifying the species, and it has been modifying also the genetic resources with largely an erosion, reduction of the genetic diversity available for the farming operation. So what are the consequences of this genetic erosion and this practice in terms of epiphytism? When we go to natural population, usually we find individuals like this in this picture. You can see a clean individual that even if we look at the stereoscopic microscope, we see that the thallus is really clean with no epiphyte growing on the surface. This contrasts a lot with the biomass produced in farms, where we usually see a large cover of epiphytes. And this is an example, probably an extreme example, but just to show you that here is the Gracilaria thallus, this black axis here. And as you can see, most of the biomass actually is epiphyte. It is not the Gracilaria. So it is really a problem in farms. the epiphytes are growing on the surface of this algae in a high density, creating a large biomass and having different types of effects. I will show you some of them in the following slides. So why is this occurring in farms and not in natural population? Well, because farms are actually dense populations. So just if we, sorry, I'll go back. If we contrast with natural population, you have usually scattered individuals with a lot of space in between. So these are low density populations. Whereas in farms, you have all the individuals touching each other on large surface. So these dense populations is really a very favorable environment for the expansion or propagation or spread of diseases, pests, and of course of epiphytes. And also you have the fact that these dense populations are genetically homogeneous. There is very low genetic diversity with the dominance of one or very few clones. And so once a pathogen arrives to this population, it is capable of infecting the first individuals, and then it can infect every individuals because they are exactly the same. So from the epiphyte or pathogen point of view, it's very favorable environment for its growth. So what is the diversity of these epiphytes in farms? Actually, there are many different species. Some of these species create like a kind of biofilm on the surface, and this is the only interaction they have with the host Gracilaria. This is the case, for example, of Archicetium. And some others not only grow on the surface, but also have interaction with the seaweed and cause some diseases to the host. Like, as we can see here with the Erythrocladia, the Erythrocladia is the epiphyte. It's a red algae, encrusting algae covering the surface. We can see it here on the picture on the left. And if you look here, the host, the cells of the host are actually decaying and dying due to the presence of this encrusting algae on the surface. So there is an impact that is similar to a disease caused by the epiphytes. So to understand this diversity, we can classify the different species of epiphytes in terms of the impacts they have on the host. From the type 1 here on the left, where it's only the presence of the epiphyte on the surface with no direct impact on the host tissues. So it is probably only providing some shadow on the host, maybe affecting the photosynthesis, but it's a minor effect. Then you have some cases where in between the epiphyte and the host, you have a bacterial biofilm that is appearing, and eventually this bacterial biofilm attacks the host and create necrosis. So this is an indirect effect of the presence of the epiphyte. In some other cases, the epiphyte is directly affecting the host. So type 3, like I just showed for Erythrocladia, there is a disruption of the cortical tissue with cells that die just below the presence of the epiphyte. And depending on the coverage of the epiphyte, then you can have more or less extension of necrosis along the host. This disruption sometimes is accompanied by the development, so here is the cortical tissue, sorry, of the Gracilaria. This is an epiphyte here. And as you can see, the disrupted tissue starts to generate abnormal growth of different cells, which is similar to a cancer, if you want. So it is really a response of the host that leads to the abnormal production of new tissue. And finally, the most aggressive epiphytes are the ones that directly penetrate within the host tissue. This is, I think it's a Polysiphonia. So it's an epiphyte, but it also grows within the host and behaves like an endophyte as well. So different species diversity, or high species diversity with different impacts on the host. Uh, these different species have different also epidemiological dynamics within the farms throughout the seasons. As you can see, some appear only during the austral winter, some others appear mainly during spring and summer, some others appear all year round. So these dynamics require adaptive measure for each season, depending on the coverage, the abundance of the epiphytes, and also on the severity of the effects they have on the host. It is really complicated to manage this the production in response to these different epidemic dynamics of the epiphytes. So we started to look at the existence of resistance, the phenomenon of resistance to epiphytes. The general idea was that when the epiphytes just live on the surface, they interact very little with the host, and there shouldn't be any response from the host in terms of response to the presence of another species. They would respond mainly when there is an impact on the growth, on the tissue, on the cells. So when we look at Erythrocladia here, the one that is creating necrosis below the encrusting algae, so we looked within farms, and actually we found some strains that were resistant to the presence of Erythrocladia. This is a bioassay where we use a clean algae and we inoculate spores of Erythrocladia. In a sensitive strain, you can see that the spores germinate and eventually develop a crust, and then appears the necrosis in the host tissue. Whereas in the resistant strains, when we inoculate the spores, they eventually germinate, but they do not develop a crust on the surface of the host, and then the cortical tissue remains intact. So there is a resistance that is possible to these types of epiphytes. Interestingly, these strains are also resistant to other types of epiphytes, like, for example, to Acrocatium, which is a filamentous algae that is a type 1 epiphyte, so creating very little impact apparently on the host. And when we look at the sensitive strains, so Erythrocladia inoculated with spores of Gracilaria inoculated with spores of Acrocatium, you can see that the sensitive strains will develop a lot of filaments of these microalgae, whereas the resistant ones will stay either with only the holdfast of the epiphyte growing, and when there is a production of an erect valley, it will grow very little. It will not develop in large filaments, as we can see in the photos on the upper picture. So there is apparently a resistance that is expressed, and it is effective to different species of hosts, of epiphytes, which makes us think that there is a general mechanism of response from Gracilaria to epiphytism. Of course, it is not universal, and we can see different examples of epiphyte species that are able to infect both sensitive and resistant strains. This is the case of Ceramium. Ceramium is a species that is able to reattach to the substratum by producing apteria. And when it reattaches to Gracilaria, so here is a transversal cut in Gracilaria, and here is Ceramium, you can see that the apteria actually penetrate within the host tissue. So it's a type 5 epiphyte, it's a really aggressive epiphyte, and in this case, there is no resistance that is working, and the infected strains can develop very large individuals of epiphytes. So there is apparently some response. It's not effective against every epiphyte. And if there are response, then we investigated what would be the possible mechanism occurring within the host that allows this resistance. So here I'll show you some results of this investigation on the biochemical side of the interaction. When we do some extracts of previously epiphytated Gracilaria, or Gracilaria that are without epiphyte, but on which we mimic the presence of epiphytes attacking the cortical tissue, so making some mechanical damage, for example. We make an extract of this seaweed, sorry, and then we incubate clean seaweeds, clean gracilaria within these extracts in order to see whether there is a response and an increased capacity to resist to the presence of epiphytes. And what we see, for example, is that it's not only the incubation within this extract that creates a response, it's the incubation, so 18 hours here is 18 hours of incubation within the extract of previously epiphytized gracilaria. But the effect is stronger when we, after the incubation, we let some time for the molecular and biochemical machinery of the cells to produce the compounds that will affect the epiphytes and limit the development. So this is, for example, these two conditions where we have 18 hours of incubation plus 54 hours in which the seaweed is growing on clean water, not with the extract. And this time was necessary to activate the defense mechanism against the epiphyte and translates into a reduced amount of spores that develop on the surface of the gracilaria. What are the compounds causing this response? We have some elements of response, like, for example, the identification of these molecules that are hydroxylated fatty acids. So the mechanism is probably the presence of the epiphytes affects the cell, which in particular the lipid membranes, which will be hydroxylated or oxidized, and then will activate other molecules or the metabolic pathways, if you want, within the cell, that will release compounds that have effects on the epiphyte itself. And when we use these molecules on the seaweed, we could see that there are indeed reducing significantly the number of epiphytes that are able to grow on the seaweed. So there are compounds that the seaweed is able to produce, and we can trigger this, the production of these compounds and increase the resistance in terms of limiting the spore settlement on the surface or the germination and development of the epiphyte on the surface of the host. Then it's more recent experiment that actually we are conducting now. Experiments in which we want to combine the role of the genetic diversity on the production and the epiphytic, the response of the host to the epiphytes. So there is one general idea that if the epiphytism is a problem that emerged from the fact that we have dense population of single clones, then we would expect that by introducing more diversity in these clonal populations, we would eventually limit the spread of the epiphytes. So we measured the effect of the genetic diversity by comparing stands with a single genotype or with 4 genotypes or with 8 genotypes or different levels of genetic diversity of the stands. And actually the results are quite surprising. The highest growth rate and the highest biomass production was on a single clone stands. This was surprising. So it suggests that eventually when you mix different genotypes, there could be some kind of competition among them for the growth. And eventually we could see that the ones that have the less epiphytic load were apparently the ones with the lowest genetic diversity also. So apparently the clonal diversity seems to be favorable, in fact, to the resistance or is limiting the spread of the epiphyte. So of course, these are very preliminary experiments. We did this with only 2 farms with very few replicates, so it's an ongoing process. But we are really interested in trying to understand how the genetic diversity actually is regulating or modulating the epiphytic load of the farm and eventually the total production of the farm. And why we think this is important? Because for different reasons. One is that this type of strategy of using single clone patches, but eventually mixing different patches within a farm of different clones, and eventually shuffling these patches from one year to the other, in terrestrial plants, this has proven to be quite effective in controlling the development and spread of pathogens. So we think it is something to be explored within the seaweed cultivation, especially when there is clonal propagation. And also because, as we saw, the cultivation of Gracilaria in Chile is largely distributed across the country. It covers nearly 4,000 kilometers of coast, so a large latitudinal gradient where the species is facing different environmental conditions in terms of temperature, of sunlight, but also of epiphytes present in the environment. The epiphytes that develop within the farms come from the natural environment, and the species that are present in northern Chile, of course, are different to the ones that are present in southern Chile. So we are trying to explore the idea that if we select local genotypes for highest growth, and that show the high resistance to the local species of pathogens and epiphytes, then we will actually be selecting land races. And this strategy probably will allow a better management or control of epiphytic load while keeping certain amount of productivity within the farms. So this requires more experiments as the ones that I've just showed you. And to finish, I just want to remember that when we try to select strains for seaweed production, actually we are developing the process of domestication. And this process is not just the seaweed, the fate of the seaweed. It is really the product of the interaction between the seaweed and the humans. The humans is selecting different types of seaweeds. The humans has different needs and different requirements and wants to produce seaweeds of certain quality or shape or flavor, whatever. But it has also some different capabilities. It is not the same when you cultivate an intensive farm on ropes with very specific requirements, as opposed to, for example, the extensive artisanal cultivation that is typical in Chile, that I showed you earlier. So that the types of farming operation, the technology that is being used is really driving what can be done or not on the seaweed. And by doing so, the human also modifies the environment. Of course, when it leads the species is being cultivated on ropes, it's a different environment from the natural one. When it is cultivated on mud or sandy bottom, it is a different environment than the natural populations. So the human is modifying the environment for the species, and this environment is also imposing some selective pressure on the seaweed. So it is really the interaction between humans, the environments, and the seaweed that modifies through generations the quality of the seaweed, and this is the process of domestication. And I wanted to remind this because when we are thinking in managing the problems like epiphytism, we need to think through generations as a problem that has 3 dimensions to be solved or to be integrated somehow. So thank you for your attention, and thank you very much to Alan Critchley to invite me and talk about these issues on this workshop on Gracilaria, bringing some, I hope, some ideas for the discussions of this workshop. I would like also to thank all the many collaborators that have been working since at least 15 years on this species. And thank you for your attention. Goodbye.